So now we're going to look at DNA replication and how DNA replicates. Now for DNA replication, you would need your DNA to make a copy of itself when your cells are dividing. So if you have one cell and it's going through mitosis, well, you would need to have copied the DNA first so that each daughter cell has an exact copy. And that's really when DNA replication is used. So when we look at how do you copy, so if you have a double helix and you need to, the cool thing about DNA, I guess you could say, is that because it's a double helix, so you have two strands, you're going to open the strands, and because DNA is complementary, if you know like this strand of DNA, you can build the complement. And over here, if I know this strand of DNA, well then I can build the complement. And then I'd have two exact copies of DNA. So the first step though would be to unwind it. You need to separate these strands, and there's some enzymes involved, of course. And so with this, there's one enzyme that is not in my PowerPoint here, but there's an enzyme that happens like upstream here, and it's called topoisomerase. And you can think of topoisomerase as an enzyme that is basically like loosening the DNA. And then we're going to use the enzyme helicase. And helicase is going to break those hydrogen bonds between base pairs. So it's going to like unzip the DNA. So then now you are starting the process of separating two strands. Now this area here, that is the replication fork. So with this, well, I should mention too, these little like green things or blue, I don't know what color they are, those are single-stranded binding proteins that are going to help, when you separate the DNA, help hold it open so it doesn't just twist back up. You think about if someone has curly hair and you like pull on their hair, it like coils back up. So here you would add these little proteins to help keep it separated. Now the replication fork though, on this side is moving in this direction, and over here it's moving in that direction. So you have the replication fork, which is where your DNA is being separated. Okay, so when we look at DNA, here you have your DNA helicase, and helicase is moving in this direction, going this way. That is the growing replication fork, and when you separate this, you're going to use your single-stranded binding proteins to help hold open those two strands. Now, it's very important to always identify where the 3' and 5' ends are of the double helix, of the two strands. Because DNA is anti-parallel, the DNA is built in opposite directions, basically. So here, if this existing strand, so I want you to keep in mind that this 3 right here is representing this strand. Here's the original. So this would be like A, T, C, G, C, A, T, T, whatever. Like there's nucleotides in this strand. So when we say it's built from 5' to 3', this is the direction that DNA is built. Because, as you saw in my other video, DNA polymerase 3 can only add on to the 3' end. So as this polymerase is going to come, DNA polymerase 3, and it's going to extend this and build the complementary strand, what we're seeing is we're going to be doing base pairing rules. So like here where there was a C, there's a G, A, T, A, T, G, like that. And so that DNA polymerase is adding the complementary base pairs to whatever is located on this original parent strand. Now on the top, we notice that since this is a 5' end and the new strand that's going to be built here, it can't be built from 3 to 5. It cannot go in the same direction as the growing replication fork. And so therefore, we have to build it from 5 to 3. So it's basically like this. As the DNA opens, you can build a section. And then as it opens some more, you could build another section. And then as it opens more, you can build the next section. So it's basically as the replication fork opens, then you could build a new section and then a new one. So this side of the DNA will be built in fragments. And we call those fragments Okazaki fragments. I'm going to go ahead and erase some of this from my slide. So now these fragments are built still 5 to 3, still by DNA polymerase 3, but in the opposite direction of the replication fork because it has to be built from 5 to 3. So again, your next fragment will be here, and then as it continues to move, then you have another one, etc. So with this, you need to seal these fragments, and that's where the enzyme ligase will come in, and ligase will build that last phosphodiester bond between the two nucleotides. So here, this strand is called our leading strand. It's built continuously in the same direction as replication fork. And then you have your lagging strand. I don't mean that to be crossed out, but your lagging strand is going to be built in the opposite direction of your replication fork. So there's limits to DNA polymerase 3, though. It can only attach to a 3' end of an existing strand. So like right here, when we just start building, it can't actually happen like that. It can't just start to attach out of thin air. So when we look at this here, in order to build DNA polymerase 3 actually is going to require primers. And so primers, okay, we're going to have to hit pause on that and put mute. So this is actually going to require primers. So there's a separate enzyme called primase, and primase is going to build primers. Now these primers, though, are built out of RNA. So primase is able to just start attaching nucleotides to the DNA, just like we see in transcription when RNA polymerase builds RNA from DNA. So here, primase is going to build RNA sequence of nucleotides that are going to base pair with the DNA. So you have some RNA built. Now there's a 3' end of RNA. That allows DNA polymerase to begin attaching and then build the leading strand. Now for the fragments, though, you also need a primer for each fragment. Each fragment needs a primer of RNA because DNA polymerase 3 needs an existing 3' end. So if you're like, what is she talking about? Down here at the bottom, here's a nucleotide. Right here, that bottom corner of the nucleotide is the 3' end. And so here's another RNA primer. Then DNA polymerase 3 can build the new fragment of DNA, the Okazaki fragment. Here's another RNA primer, etc. So in reality, you need to have a primer first in order to allow DNA polymerase to build the complementary strand of DNA. But the problem here is that you can't leave RNA mixed in with our DNA nucleotides. So we're going to need a way to actually remove these RNA primers. So a primer is built by primase, and it serves as the starting sequence for DNA polymerase 3. But now we got to go in and we got to take out those RNA primers and replace them with DNA nucleotides. And this is where DNA polymerase 1 will actually come and remove these RNA primers and replace them with DNA nucleotides. So DNA polymerase 1 will take out the RNA and put in DNA, and then ligase will come and seal those phosphodiester bonds between the sugars and the phosphates. Now, up here, though, at this first one, you had RNA, so it gets removed. Here is the problem. This is the 5' end. DNA polymerase 1, DNA polymerase 3, they both cannot attach to the 5' end. So every time your DNA replicates, your chromosomes get a little bit shorter. This end will not be replaced, and this is called chromosome erosion. So DNA polymerase 1 replaces or removes sections of the RNA nucleotides and replaces them with DNA, but it can only add on to the existing strand, and therefore our chromosomes will erode as life goes on. So every time your cells divide, your chromosomes get a little bit shorter. Now, thankfully, we have telomeres. So telomeres are at the ends of our chromosomes, and they are like your aglets or the tape at the end of your shoelaces. It's repeating sections of DNA that don't code for genes Therefore, if they get eroded, if they get like shorter as time goes on, then you're okay because they're not coding for anything critically important. It's when your telomeres are gone, then you start getting into your important sections of your chromosomes. That's when the body begins to shut down and people can die of natural causes. Now, they have found that diet actually plays a large role on your telomeres. The more plants that you eat in your diet, the better maintained your telomeres are. We have this enzyme called telomerase, and telomerase will keep your telomeres there for you. So cells that are actively dividing all the time would have high amounts of the enzyme telomerase to help maintain your telomeres. But we could talk more about telomeres in class. Okay, so they extend, or the enzyme telomerase will extend your telomeres, trying to keep them there for you. So that way your DNA can continue to divide for multiple generations of cell division. And there's different kinds of polymerases. DNA polymerase 3 is going to be our main builder, building that complementary strand. It's about 1,000 bases a second. And then DNA polymerase 1 is going to be the one that removes the nucleotides and replaces them with RNA. And it also searches for mutations and tries to correct mistakes that it comes across. So let's go ahead and summarize here. So this is the direction of our replication fork, which is opposite of what we saw in the first slides. So here's our helicases, green. Our single-stranded binding proteins are going to hold open those two strands of DNA. Up here, the pink enzyme is primase. You can tell it's building the RNA primers to build the Okazaki fragments. Okay. Here we have DNA polymerase 3 is orange. You can tell that this strand on the bottom is our leading strand because, okay, there's our leading strand because it's built in the same direction as the replication fork. And up top we have our lagging strand. It's built in the opposite direction of the replication fork. We also have our DNA polymerase 1 right here that's going to replace and take out the RNA nucleotides and replace them with DNA nucleotides. We have our ligase. Our ligase will seal the sections between the fragments. And this is replication. All right, good job.